Dear students, today I'm going to explain a little about the recombinant DNA and its social responsibilities. Back in 1918 won Nobel Prize of the DNA on the DNA cloning. Uh, After that achievement, scientist Andy Berg himself was worried about its use. Berg arranged the meeting of the 100 molecular biologists. After this meeting, NIH formulated the guidelines. uh, what were the issues that were that were in his mind, and what was the problems behind these all achievements. There were some issues regarding the biological containments, regarding the use of such organisms. Means biological containment means the development of the vectors in the host cells that cannot be manipulated outside the labs. There was the high risk work. The high risk of work was done for the modifying the vectors, because the animals are manipulated, animals are modified, so the risk was for their misuse. Acceptability of the GM crops. Current concern in the GMOs, genetically modified organisms, uh, was one-fourth of the American land is cultivated by the GM crops, mainly for insect resistance. These modifications reduce the insecticide use, but these have severe effect on the environment. Severe effect. What kind of the severe effect on the environment was many valuable insects was also killed because of these GMOs, and it was definitely by the mistakes, because when the insecticide resistant crops are being designed, so the threat of valuable insect killing was not their mind. Because of these threats, demand for the unmodified crops is increasing. Ethics must be considered on human use. The recombinant DNA dispute is now a conflict between the academic freedom and the industrial secrecy. There was also the medical related issues. Technically, human genes grows older with the age. It's feared that the clone individuals would be retained the age of the donor's gene. So the donor genes will obtain its original age when it is incorporated into the some other organisms. The inoculated egg and the transferred nucleus may not be compatible. Uh, an egg with a newly transferred nucleus may not begin to divide or develop properly. Implementations of the embryo into the surrogate mother might fail. The pregnancy itself might fail. So there was a lot of problems with the organisms. If we go towards the uh, artificial generation of the organisms, if we go towards the cloning of the animals, cloning of the animals through the somatic cells nuclear transfer is simply inefficient, The success rate ranges from the 0.1% to 3%, which means that for every 1,000 tries, only 1 to 30 clones are made. Problems during the later development, these were the problems when we establish the reaction between the egg and sperm. This is the initial problem. These are the initial problems, that the pregnancy might be failed, that embryo cannot be incorporated in or implanted into the surrogate mother, or pregnancy itself may be failed. So if the pregnancy is established, then what kind of problem will be happen during the later developmental stages? Cloned animals that do survive tend to be much bigger at the birth than their natural counterparts. Clone with the large offsprings syndrome have abnormally large organs. This can lead to breathing, blood flow, and other problems. So because clone genes, as I already had said, that it may obtain the age of its donor. So there may be the abnormal gene expression pattern can also be observed in such kind of organisms. Telomeric differences can also be found as cells divide, their chromosome get shorter. The telomeres length of the cloned animals found no clear answers. Chromosomes from the cloned cattle or the mice had larger telomeres than normal. These cells showed other signs of youth and seems to have an extended lifespan compared with cell from a naturally conceived cow. Recombinant DNA and the social responsibility for this conference was held by, as I already said, that more than 100 scientists to figure out what kind of social responsibility must be on the humans, must be on the scientist. Good judgment key, the NIH guidelines will never be complete or final since all the convincible experiment involving the recombinant DNA cannot be foreseen. Therefore, it is responsibility of the institution and those associated with it to adhere to the intent of the NIH guidelines as well as to the specifics. According to the NIH, if a scientist want to work on the recombinant DNA, recombination of the DNA, the scientists must follow the guidelines that are made by the WHO or that are made by the NIH. And there must be also a specific guidelines for made by their institutions. Scope specific practices for constructing and handling recombinant DNA molecules, organisms, and viruses containing recombinant DNA molecules. Um why these guidelines were made? These guidelines was made to secure the life of the scientists, secure the life of the organisms, and secure the life of the environment. Um, Are the NIH guidelines are optional? Optional? No, it doesn't mean that these guidelines are optional. If you work with the recombinant DNA, you need to follow these guidelines. Uh, what are the potential consequences of non-compliance with the NIH guidelines? Suspicious limitations or termination of the NIH funded projects will be done. If a person who got the funding for the recombinant DNA technology from the NIH, if the scientist is not following the NIH guidelines, his or her project will be cancelled. His research will be stopped. Institutional responsibility. Institution shall establish and implement the policies for the safe conduct of the recombinant DNA technology. Institution must have a biosafety committee. So this biosafety committee will be responsible for the uh, proper proper following of the NIH guidelines. Governmental responsibility, it is the responsibility of the NIH director to establish the NIH guidelines and oversee their implementation and finally interpretation. Similarly, the government is also responsible to look on such kind of the projects. University responsibility, it is the responsibility of the university to ensure that all the recombinant DNA technology, recombinant DNA and the synthetic nucleic acid molecule research conducted at or sponsored by the university complies with the NIH guidelines. General responsibilities of the PI, principal investigator, must initiate or modify, must consider before the initiating or modifying DNA or synthetic nucleic acid molecule research subject to the NIH guidelines. Only after that research or the modification has been approved by the IBC. Until or unless the project is approved, he must not initiate the recombinant DNA project. Being adequately trained in good microbiology techniques, the scientists must be trained, must be good in microbiological techniques. The scientists must make the protocol available that describe the potential biohazards and the precautions to be taken. The scientists must inform their staff of the reason and the provision for any precautionary medical practices. They must consider the guidelines and they must consider the life of their workers. So this is all about this is all about a little bit about the social responsibilities, including social responsibilities to safeguard the life of the organisms. If we look on this document, we can divide the responsibilities in almost three levels. One is the governmental level, or you can say one is the responsibility based on the funding agency, and second responsibility is based on the institution where the research will be conducted, and third is the PI. Third is the principal investigator, the scientist who will conduct the research. The responsibility of the institution and the funding agency is to make sure that the scientist is following the NIH guidelines, while the principal investigator is responsible to provide the environment according to the NIH guidelines. Hoping this will be easier to understand, we will meet tomorrow with our next lecture. Thank you, students. Hope it will be easier for you. Thank you. Allah Hafiz.